Satan is abroad in land, and he goeth forth deceiving the nations. Wherefore he that prayeth, whose spirit is contrite, the same is accepted of me, if he obey my ordinances. I promise you, you are known as covenant-making and keeping children of our Heavenly Father. You are known, and it doesn't make you more loved than others, but because you have made covenants, it accesses certain blessings that others don't have. One of the greatest treasures is the opportunity to have the gift of the Holy Ghost in our lives. And I hope that you're learning to access that more and more. However, the Godhead be with you at all times as we keep the commandments, to teach us truth, to illuminate our minds, to help us remember things. That's powerful, you guys. It's really powerful. So please learn to use and access that joy. Okay? So you're not deceived. A lot of fun things up here. Let's look at it from the standpoint of the study guide. Okay, so we're going to get 2 days on chapter 8, and it says we're supposed to, there's that word, memorize the structures and names of the nucleotides. It's okay, this isn't going to be too bad. We'll practice a little bit today in a way to show you at least an outline that will get you some of it going down. And the reason why is because you'll need to draw a short single-stranded polynucleotide. So on the right arm, you'll be asked to draw a short strand, phosphodiester bond, link it, and it will either be DNA or RNA, and you'll be fine. Be able to distinguish between a purine and primitive. Check. What's the structural differences between ribonucleotides and deoxyribonucleotides? Describe the proper Watson-Crick base pairing. Why does DNA exhibit a major and minor groove? Describe the level of conformational flexibility of the polynucleotide chain for DNA versus RNA. Yeah, you guys are. You've also got 3 types of double-stranded DNA which are relevant for it. What features of RNA allow for more structural diversity than not seen in DNA? What structural motifs contribute to the tertiary structure of RNA? What are some important physiological and chemical properties of the nucleic acids, and how can they be changed? What's the relevance of DNA regions that are rich in DC pairing? And that will get us through chapter 8. So chapter 7 is going to have the bulk. Chapter 8 is definitely going to be we'll use both chapter 8 and chapter 9, but it will have more in chapter 8 than it will be from chapter 9. So it's going to be the lightest section when we go through 9. Okay. When's the test?
Next week.
Next week on Friday and Saturday. Okay. Well, it gets us back into where we want to be now. Nucleotides, are they only for coding for our genetic information? Yea or nay? Nay. Well, at least the ribosome's a enzyme. So that's actually helping turn it into it. Okay. We also have cofactors like our NADH, our ATP, our cyclic AMP. These are all different things. So FAD, NAD are redox cofactors, and they store energy, and they're made of nucleotides, nucleobases. Signal transduction we'll talk about as well in the next unit, but cyclic AMP, some of you, or CAMP, others are going, oh, I know that one. Yeah, I got it, right? And most of us have heard of ATP at some point in our life. It gets thrown out there like it's candy. Yeah, it's good stuff. Okay. So obviously genetic information also involved in protein synthesis, our tRNA, our rRNA, messenger RNA, good things. So top level, a question on the test. Boom, right here, are you gonna get it? Do you know the difference between a nucleotide, a nucleoside, and a nucleobase? No. Everything you need to know is right there. And I'm going to go with the one has a phosphate on it. the other nucleoside just has the sugar in it, and then it's just the actual nitrogenous base. And with that said, if you've marked that correctly, you are one less multiple choice that you'll be missing. Don't get it right. Okay? When we look at the phosphate, it is attached at the 5' end. That will become important when we start talking about how we actually transcribe. We go from the 5' to the 3'. 5' end right here off of the 5th carbon, 3rd carbon, and that's actually how we will be linking them together. So when you go later to create your phosphodiester backbone, you'll have the phosphate, and as you do, you'll be linking directly to a sugar with the ribose ring, and you're like, oh, yeah, I'm gonna put it at the 2, and obviously this will be up at the 5'. Sorry, bad drawing day. And then we'll just keep going right down on our next one that we're doing, okay? And you can keep going right down the chain. So you've got to be able to do that, and then you will be putting off on it the nitrogenous base as well. And so at the first start, they're all going to be beta. Remember I told you you can't decide whether it goes alpha or beta? You can't. Enzymes can control things. And they want beta. Okay? To our pyrimidine or purine base. There's AMP. There is cyclic AMP. There are other ones where the phosphate can get attached as well. Just know that it exists out there. What's the difference between DNA and RNA? Tell your neighbor about it. You're smart.
Thanks, Chad. Deoxyribose is DNA. RNA is ribose.
Can you imagine somebody drawing down, and I give you off nitrogenous bases that say it's RNA, and then you put it as if it were DNA? Somebody will do that. Don't let it be you when you're drawing your structure. Right? That would just be painful, open admission that you're not paying attention. Right? Okay. Where did our proteins pick up light? When we got our resonance structures of our aromatics, where were they absorbing light? What wavelength? So 260, and what amino acid absorbs at 260? The phenylalanine, okay? But that was the lowest signal. What was the major signal absorbed at? 280, and that was the tryptophan for most of it, and then also the tyrosine, okay? So you should know that, and it turns out that DNA and RNA absorbs at 260, which means that when I'm analyzing DNA, which has been purified out of some cell, I'm going to also be looking at the 280 signal as a ratio, knowing that I might, if I didn't get it cleaned up enough, that I might have also contributions coming from the phenylalanine. So I'm not going to believe it's 100% pure. So most automated instrumentation that does it automatically, like nanodrops that we have several of in the other room, they'll do that for you automatically. Say, ooh, you're this purity. They'll look at those together. Okay? So that'll absorb UV light. They do have structures like that. So there, cytosine, DNA, and RNA, thymine, only DNA, uracil, only in the RNA. Let's see, what's the difference between uracil and thymine? A methyl alone. In fact, there's an enzyme that's actually called diamidine methyl synthase. And yes, what does it do? It adds the methyl to uracil. That's just a role of one enzyme. That's how it's built. So they look identical, so that's pretty good. Do you notice these are largely the same as well? What's the real difference here? The amine here, which meant I also have this one deprotonated, and obviously beyond that, just depends on which one I'm looking at, right? But that's really fairly straightforward. So you guys, it's worth your time to take a moment on your paper and to practice drawing one of these. And we're gonna pick on this one because it seems to be the most in common. So we'll draw uracil, and in drawing uracil, I'm gonna draw this down. I've got my double bond here, right? Okay, fantastic. I know I'm a cyclical structure. Turns out that it's a nitrogenous base, so I've got a nitrogen here, and then I've got my carbonyl sticking off this guy. I've got another nitrogen that's gonna be on my corner, and at the top I'll go with the carbonyl again, okay? Now that's a pretty good start on dealing with learning, okay? So how many nitrogens did I draw? Two, okay? 2 nitrogens, both of them protonated. How many carbonyls? 2, also next to each other, and then the double bonded group. Seems reasonable so far, right? So practice these things so that it becomes easy. It's not as hard as you think. Now I want you to keep that on your paper, because when we now look, that was our pyrimidines. When I look at the purines, I want to see if we have anything in common. Did my nitrogens move position? No. How about the carbon double bond? No. In fact, this carbonyl stayed in the same spot. If I'm looking at this guy, wow, this is so similar. They are similar, okay? Not accidental, has to do with the building blocks they're constructed from. But it means in this case that coming over here, I'm gonna turn around and pull off the hydrogens and draw in my nitrogen and another nitrogen in this position, and then I'll do a double bond. And so that's feeling pretty good so far, and then I have to alter just a couple things because this one's gonna end up being an amine group, right? And because I pulled that down, I also end up with this, which deprotonates this guy, and I think we're pretty good. Okay. So minor changes. Learn the backbone of your purine and your pyrimidine, and what are the minor changes that you need to do? It should be minor things, okay? Not major. That will make it easier for you to deal with. Is there a reason they call them nitrogenous bases? Yeah, yeah. So how many of you, so when we talk about acids and bases, acids, you know, taste sour, right? So you've probably tasted lemon juice and vinegar, and you're like, okay, I get it, that's sour. Bases, there is a base that we typically taste in our life, and it's one that most of us actually taste and then go, ooh, that is bitter. How many of you tried just raw cocoa powder because you thought it would taste sweet? That's a base. In fact, it's a nitrogenous base. And we'll discover later it's one of the forms that's out there. So, you're just like, oh, cool. Caffeine, by the way, is also a nitrogenous base. Oh, fascinating, okay? So they're neutral at pH 7, okay? This is important in looking at, now this is essentially the backbone, but I actually find it easier rather than throwing your curveball here and having it move, to start with one that you memorize that's actually one down. Rather than go here, okay? But this one's numbering them accordingly, and the 1 position and the 9 position are important. Why are they important according to what's ingrained?
That's where bonding happens.
Bonding to what? the ribose sugar, which do you have to memorize that one already? Yeah, and I think you guys can handle a phosphate. So in that sense, the ribose is already done, you just need the nitrogenous bases, and you are golden. And you could draw me which one was the nucleotide versus the nucleoside? Which one had the phosphate? The tide had the phosphate. Okay, good. All right, so this is where I link to the sugar and it's in beta configuration, okay? Off the anomeric carbon. Okay. I'm gonna ask a question. I'm gonna ask it in a joking way, but is it a good idea to sin? No. It probably is a bad idea. So, it turns out that the textbook tends to draw them, and many people draw them in sin. Which is still, though possible, it's sterically restricted, so we would rather go anti. So we'll be a little more like the anti-E by Lehi's here, right? Sound good? So make sure you know what I'm talking about here. This, not so happy. This, much happier. So as you go to memorize them, draw them this way. In my notes, I'll have versions that are drawn that way for your viewing pleasure. But if I draw on the textbook ones, they almost always draw them in sin. They really do. Because it occupies less space. Okay? All right. The tautomerization, I wouldn't be too worried about it. Just need to know it happens. Cool. Awesome. Moving on. We already talked about absorbing light. What was our wavelength? 260. What would be a reason to remember 280? Because in context, that's one that somebody could ask questions and say, which one? So no 260, but also know the 280 and why. Okay. Yep. Cool. I hope you know what a highest occupied molecular orbital versus lowest occupied molecular orbital means, but we're not going into that today. But this is definitely another test question. In addition to learning to draw these, not only do you need to learn what nucleoside and nucleotide are, it would be a really good idea if you learned something about the way they're named, okay? So let's see if we can find any patterns whatsoever. The nitrogenous bases, you're gonna memorize their structures, so you're pretty much set there. And notice these ones end in the "ene" ending, but that doesn't help me a great deal. But what do I notice about the tide endings? They have the 8. So these guys have the 8 ending. So when I have the phosphate on it, it's got the 8 phosphate. These also have the 8 ending. Great. These ones are the sides, so they've got the sign, 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 sign. Now this is where it messes it up on me. But yeah, they're the ones that also are there. Yeah, question?
So on the study guide, when it says memorize how to draw like all the nucleotides, are these all the nucleotides that we're required to? Okay, cool.
Yeah, they are. Okay. All right. And they don't just come by full names. It turns out we also have them in, shocker, 1 letter, 2 letter, and 3 letter, 4 letter, or 3 letter forms. It's really not that bad. Most of you actually know them as if I say A, C, T, U, you're like, the 1 letters were easy. Just don't ask me to spell the word. Great, it's multiple choice. The spelling of the full word is not there, but recognizing of the name being a phosphate or a phosphide, that doesn't, those are what you need to know. A nucleotide and nucleoside. So again, I drew these in anti, right? And they're the deoxy forms, so that you're set there. Which also means this little d means I'm looking at the deoxy form. And here I'm looking at the ribonucleotides, so I've got both of them with oxygens in these positions, and now I've removed the little d in it. And I'm now working with, what was the one that was here that's not in the other set? Uracil or uridine, yep. And then in the other one, what was it? Thymidine. And what was the difference between the two? A methyl group. fascinating. Cool. So, yes, it takes me 2 seconds to show you a slide that for some of you is painful, right?
Yep.
Yeah, you're gonna be all right. You're gonna make it, okay? Almost there, okay? All right, what do we need to discuss here? Polynucleotides, phosphodiester linkage. And so we'll look at that. Why do we call it phosphodiester? The RNA, though, is known as being unstable, and DNA is much, much more stable. Now, they believe when life originated, it was actually more of the RNA, but it's unstable. Any idea why RNA is unstable compared to DNA?
One side is exposed.
One side's exposed, okay? It's a common answer. What do you got?
You've got one more functional group that can do reactions.
Okay, that's true. That's true. Yeah?
Actually, never mind. I was thinking about locations of them, but I don't think that really comes into play that much.
Yeah, they both move in the same locations
Yeah.
within the cell, so that's not it. So the most common answer given, if I were to ask students that haven't taken a biochemistry class, will be RNA is single-stranded, DNA is double-stranded. And that is wrong. So forevermore, that is not your answer. However, you can see why they would go there, okay? But let me ask you first a question. Is RNA ever found in double-stranded?
Yes.
Yep. Is DNA ever found in single-stranded?
Yes.
Yes. And it turns out that RNA, whether it's double-stranded or not, doesn't change this ability, nor with DNA. It has something to do with what you were mentioning, Glenn, the functional groups, and there's actually a spot where it's reactive. And this will show up on the right arm, okay? So we will talk about that, but here's a picture of the phosphodiester backbone. So there's really a phospho with an ester linkage here, and a phospho with an ester. That's why they call it the phosphodiester when I've made the chain, okay? You don't get away with just writing G or C. I should have to put in the nucleobase, sorry. And draw, I mean, which way? Sin or anti? Oh, you guys are awesome, okay? So that's good. And we will talk about base pairing here. You always draw them 5' to 3'. So when we learn to write proteins, where were we going from? N-terminus or C-terminus in the way that we wrote the letters? N-terminus, and then we wrote to C-terminus. So we started on the left with our N-terminus and we went to the right, just like I write words. That's great. How do you think we do it with DNA? 5' to 3'. And that's really, so we're starting at the top. And so the way that it's written, ATG, means I should have had the A at the top, TG. Okay, same with my RNA. Okay? All right. There are enzymes that will break down RNA as well as enzymes that will cut up DNA, by the way. But hydrolysis is the name of the game. If I have DNA, I don't have this hydroxyl. This hydroxyl here, they're showing it in the cheat way that they like to show it, which is that these electrons, instead of the lone pair, are attacking the phosphodiester bond. But essentially what happens is you get a water that comes in. It's usually a slightly alkaline, but even at neutral pH, I'll have some hydroxides that'll be there. These have the ability to then come in and attack and deprotonate that the hydroxyl here at the second position. When that occurs, it's instantly electron rich, it's negatively charged, it says I gotta find a home, and already in the right position, really close to it, is the phosphodiester bond with this big scream of saying, please come attack me. And it does. And so the electrons hop over here, which then says, I've got too many electrons here, so I'll break the bond, and actually that cleaves and pulls apart that phosphodiester. So inherently, the instability. Now, what do I need on the exam? This. Okay? Now, by default, you've already created the phosphodiester bond. In fact, you will have drawn something already. So really, all you need to learn is 3 arrows with an OH. You don't need to draw this part. You're welcome to, but 3 arrows. But it will show up. But if asked the question, why is it unstable? You're going to say, well, it's not because it's double-stranded or not. You're going to say it's inherently because the functional group of the OH allows for a nucleophilic attack with a hydroxyl that will then attack the phosphodester, and then you can draw the mechanism, which is really what you want to do. 3 arrows. Something you actually will already have on your page. Okay? Easy? Okay. By the way, I say it because it's valuable so that it sinks in on you, but I really have been given permission. Brother Rubish, Brother Lemon, and I sat down and decided that it's so important to know the difference between why is this, because it's a biochemistry principle. They decided I could flunk you if you miss it on the test. I have an A, and now I don't. So don't miss it. Would I really do that to you? Probably not, but don't press it.
Challenge accepted.
Okay, just put down that you know what's going on. All right? Don't get me into this single double-stranded. That's just not where you want to go. All right. Is it obvious A pairs with T and T pairs with C? Do you wonder why? It might be nice to look at hydrogen bonding. Let's look at that. Ah, beautiful. Hydrogen bonding, we got this carbonyl, oh, amine group, that's really nice. Oh yeah, this amine group, oh, an acceptor there, this is really nice. Distance, space, just right for hydrogen bonding. And this is how we do base pair matching. Watson and Crick didn't know this is really pairing my thymine, right, with my adenine, and then over here my cytosine with my guanine. What do I notice about this one versus this one?
Cytosine guanine has more hydrogen bonds?
Yeah, I get 3. So what do you think about the energy required to pull apart regions that might be rich in GC? Much harder, right? So if I were throwing this into, for example, Yellowstone in a hot pot, and I wanted to survive with the DNA, would I choose to put a lot of AT, or would I choose to put a lot more GC in it? GC. Probably much richer in GC. But that said, I still have to code for proteins, right? Yeah, so it's not an exclusive thing, but that is true. That's really what happens. Okay, now, another question will inherently come up, and I should totally require it, but I don't. Will I have to draw the hydrogen bond? You should know it does, and you should definitely know the GC gets a triple bond and the AT gets a double, and the hydrogen bonding, but you won't be asked. One might ask, why is there not a hydrogen bond between these two? And you can answer that to your neighbor. Please tell them why the thymine to adenosine. Why do I not have one right here? This is just a check for understanding of hydrogen bonds.
The adenine, the hydrogen that might bond is connected to a carbon.
And somebody try to explain it to me. What's going on here? Was it the carbonyl?
Nope.
No, what was it?
The hydrogen is attached to carbon.
Which the difference in electronegativities is small enough it's considered non-polar. So definitely not a hydrogen bonding, large electronegativity difference dipole that's one of our set. Okay, so that's why we're talking about it. Okay. Are there minor nucleosides? Absolutely. Okay, methyl groups get thrown on. They became something involved in epigenetics. Have any of you studied enough genetics during epigenetics conversations? No? You guys just gotta get a life. Okay? So epigenetics is kind of cool. When we come in and we add in minor nucleosides, something like these methyl groups right here, there are enzymes that will do that. No different than in our paper we read about cancer where we could have methylated the histones. Well, it turns out that when I start putting methylations on, one of the classic stories that's told is about a rat. A rat mother licks her pups versus doesn't lick her pups. There's implications that are astounding. The rat pup who gets licked gets to hang out with all, meaning literally her tongue licking, not beat him up. But that rat pup will usually grow up acclimatized and deal well with other rats and will live with the rat pack. And it turns out that we can observe differences in epigenetics of methylations, whether or not she does or doesn't lick the rat pup. On the other token, if the mother chooses not to do that, the rat pups will end up growing up more aggressive. They tend to not be able to live with the rest of the rats. They end up being isolationists, and their survival rate is thus also less. Okay? Now, let's just draw an implication to you. If your mother licked you, okay, then you get to be with society. And if not, you get to be an outcast, right? Isn't that a great idea? Don't go lick your children, but did you listen to a general conference just a couple of years ago about a bunny that the bunnies live longer if they took care and loved and held the bunnies? Same implications, right? It turns out that when you love your children and you give them physical attention, it actually makes them better. Yeah, really.
Doesn't that kind of probably equate to like how they always talk about how important it is right after birth for the child to rest on the mother?
That's true, and we will talk briefly. I usually talk in my second semester about what goes on with that, but the hormones that are involved in that turn out that they're the pair bonding, and they're rich when you've actually just given birth and also when lactating. And it goes both the mother and child. So that produces an extreme pair bonding, which later, what I would mention in that same vein is for guys who manage to figure out as husband that you should show up when the birth is happening, it's a win, because all of a sudden, your wife, who has got all these hormones going through her, is likely to look at you and go, "Oh, I love you." Even though just 2 seconds ago she was going, "I hate your guts, and you will never come near me again." And that's very true, okay? But it'll switch the tables because you'll be part of the pair bonding and life will be all good for you, okay? So, guys, did you learn something? I think that's a good idea. You're thinking, I will be gone out of town when the baby's born. You missed out. You're going to regret that. You really will. Okay. And gals are taking note. Note to self, don't love it. Okay, so these are real things that occur. All right. So there are other epigenetic implications. I'll give one other quick one, then I'm going to go forward so we don't get too far behind. One other very famous epigenetic study is actually given by a small group that's out of Sweden, and then they were in a very northern country, and the city or the little town that was there kept their records for well over 100 years of all births, all deaths, and also the crops, and whether they were famine years or good years of the crops. And there was very little transit in and out. It turns out that when it was years that there were really poor crops, that it actually changed the females who were being born that year, that their children because that's when the eggs are developed, is when women are in the womb. Their children have a high risk of diabetes. A substantially high. I think it's like almost 17-20% chance. High. Now, then in years where they have enough. Now, if you caught that, that also would have implications that when you decide to have a child, you're gonna think like some of the women that I worked with in my graduate school, one of them actually ran not a half marathon, a full marathon within the last month of her pregnancy. Now that's insane, okay?
Wow.
And I had another gal who was in my ward, and she's a very good friend of mine, and she was a dancer. Now this was just weird to me, but I was good friends, and I was a scoutmaster. That was the most odd and disturbing sight ever to walk into as a dancer. She's a ballerina. She was doing full standing splits at 9 months pregnant. I'm like, that is not possible. And it's just, that's just a sight you don't want to see, I'm telling you right now. Like, wow. But guys, if they had a year of plenty, and it was assumed that they were obese because they had multiple years of plenty, and that's actually what they equate it to, and they've checked on. Turns out that entering puberty, which is when the sperm mature, if you are obese, you increase your posterity's chance of having diabetes by a substantial factor. So if you have children, and especially as then mothers and fathers, and you're thinking they're playing video games as they're entering puberty, what do you say to your boys? Get out there! Get out! Go do something, run, exercise, do something, right? Because you're saving your grandkids.
You sit them down and you go,
Okay.
can you see this?
And it goes, by the way, epigenetic changes tend to go to the 3rd and 4th generation, just like the scriptures describe. Drugs, by the way, smoking, all these things have effects on epigenetics. So, fascinating little things. Okay, DNA codon chart. We will come back to this at the end, but there are, if you are new to the world of biology, you use 3 nucleotides in your RNA that after you make a copy, they will actually translate into amino acids. We will practice at the end of this unit doing that translation, where we'll transcribe and translate right into a protein, okay? And so we'll want to do that. And there is something known as a start code, the AUG, which is a methionine, by the way. And then we'll also look for the stop codons, which there are 3 of them. And how do we find that? So it turns out that what we've discovered is that everyone who thought they knew it in biology, we've actually discovered about 50% of them think they know it and don't. So we tend to practice it at least one time. Yeah. Good times, you'll love it. All right, now I'm going to bring up this over here. Do you notice that actually these right here are redundant, right? Well, some of these have a different frequency that the codons are found out in nature. And it turns out that beyond that scope, we have learned that that frequency, I'll just cite the research really quickly, I don't remember where I put that, but I'll probably run into it. We have learned that, I'm sure I've got it somewhere here. Oh yeah, here's your mutations here. Mutations where it actually is producing this very same amino acid, but the mutation was one of the lesser frequently ones, actually tends to have that protein coded for less, producing less copies. It alters the expression. We thought they were not a big deal, but we have hence learned that's not true. So in 2023, we started to actually realize this. We've actually realized it for a couple of years before that, but we are really starting to understand that a lot of diseases are caused by these silent mutations, that it actually produces the same protein, and it's still produced, but they're not as likely. And that can cause discrepancies in how things are expressed. fascinating. So things that we didn't know. Now, there's also something that's part of that which allows for this base pair, which is the wobble position. So the first two matter greatly. The last one is known as the wobble position, and we can actually have a mispair. Now this is actually where my tRNA is lining up with the messenger RNA in the ribosome to find the amino acid it will code with, and that last wobble position can actually move on us to have different options. So that's kind of a little fascinating thing. There are some base pair also substitutes like inositine that also get put in there as well. Okay, so there's our inositine. There's also a pseudouridine that are found at that curve. Okay, you didn't see a lot of green on that, did you? Still interesting. All right. There are other modifications that can occur in unusual positions, putting the ribosome on in not the normal position. Where should it have been linking here? What number? One, which would have been to the nitrogen. And on the other one,
9.
at the 9th position, is that also a nitrogen?
Yes.
Yes, it is. Okay, so they're always to the nitrogen, beta, okay. Yeah, methyl groups, sulfur groups, deaminations, which we'll talk about. Here's some other infrequent ones but are interesting to hit on. Most of you know caffeine. Some of you know it way too well. In our next unit, we will talk about caffeine and its implications on you, as we'll study it so you can make some reasonable decisions like we do with our alternative sugar substitutes. These are actually the xanthine class of nucleotides. Xanthine itself is always used when we break down our nucleobases. It's actually a product that we use. But these are all xanthines. Theobromine, theophylline, caffeine, xanthine. When I would get the cocoa powder in the natural product industry, I would test it and I could tell you how many of each of these is in there. Turns out that cocoa, dark chocolate, is known to be healthy for you. Isn't that like the best news ever? Like, oh, it's healthy for you. And the reason why is actually stemmed in this theobromine. What does it do to you? Decreases your risk of cardiovascular disease, reduces your bad cholesterol, improves your good cholesterol, improves your blood flow, and supports your heart. That's great. And it turns out that there are different amounts, depending on where you came from, of that theobromine, and it actually has to do with the country it came from. Why would it alter that amount based on the country it came from? Temperature and humidity changes the way that the beans grow inside of the plant. It affects it, which, by the way, it would for coffee too, but that's irrelevant. Kind of interesting, isn't it? So now you're thinking, where do I get my chocolate from? That's a big difference.
Coffees, different places have more caffeine.
Yes. They do. Okay. And that's why the darker the chocolate, the better for you. All right. So Watson, basic crit. These guys are given credit with the Nobel Prize, and here's sort of the brief history, and then we'll end here. Structure of essentially DNA in 1929. We didn't know very much, did we? Structure by 1935. We've now got it attached to the ribose ring. We've got the phosphates there, and we know their bases. Okay. This is in 1940, a fellow by the name of Ch8, and he comes up with a connection where he says the amount of A I have is always equal to the amount of T, and the amount of G I have is always equal to the amount of C, and it turns out that's true. So ironically, in the last 2 minutes of class, we're going to solve this problem, which is and will be a great question for the upcoming exam that you'd like to get right. So let's see if we can do this. There's actually 2 different ones here. I'm going to give you only one. So in species, X of bacteria and Y, the adenine makes up 32% and 17%. So if we're doing X, it has 32% of adenine. So what are the other three? Tell your neighbor, talk about it with your neighbor.
T is 32%.
Once you got that, then have them solve it, you solve it, the second one yourself. You're thinking, bring it on, ask a lot of those questions on the exam, right?
No, not T, it's C. It's right there. Don't worry, I tutored this.
Species X, you do all right?
Yeah.
How about species Y? Check. All right, we'll come back and we'll deal with things like major and minor grooves. You want to get ahead of the game, obviously do the ones that I know, but make sure you get your extra credit in. It will be worth it. You want to do that. Yeah, you're good.
I mean, not the laptop.
Ready? Check this out. I hope you look out for this little sweet treat on the YouTube video.
It's on the YouTube video.
Chat. Matt needs an iPad.